We can see your screen. Thank you.
Wonderful. Okay. All right. Well, thank you very much for having me. I'm excited to share my shape analysis software with mycologists. I think it has tremendous value to your research because in all of biology, cells and tissues and molecules behave the same rules, whether you're talking human disease, which is my expertise, or mycology. And so today I'm going to talk about how ShapeGenie works and provide a standardized way of, or suggest a standardized way of photographing mushrooms, right, for shape analysis. I look forward to your feedback and your future contribution as to how to refine this, since you are the experts in mycology. So the organization of my talk will be as such. There'll be 5 sections. All right, first I'll talk about how ShapeGenie measures shapes. Then I'll talk about why we study shapes in nature. And then the 3 general categories of mushrooms, according to me, a non-mycologist, right? And then 4 recommendations for how to take pictures of one category, that is the pedunculated mushrooms. And lastly, I want to talk a little bit about how ShapeGenie can measure fungal spores and reveal insights that are hidden to the naked eye. So to begin, let's talk about how ShapeGenie measures shapes. So ShapeGenie, okay, it gives you 1 or 2 options. What it does is in the top row, you have a crescent moon shape, and the bottom you have this enclosed shape. And ShapeGenie allows you to overlay 1 or 2 grid systems, so either the parallel grid system or the radial grid system in the bottom. And the purpose of both grid systems is to find intersections between the grid lines and your object, as you can see in B and D. And once it does that, then it can calculate the distance of every intersection to the reference point. For the parallel grid line, the reference point is a vertical line at the left of the image. For the radial system, the reference point is the center of the bounding box, the red dot in the image. Okay, so the goal is to find intersections and calculate distance from each intersection to the reference point. And once it does that, okay, now you can turn the data into what looks like step 4, right? That is step 4. All right, it's just pairs of X, Y coordinates. X coordinate is the grid line position, and then the distance is the Y coordinate, right? And so essentially, step 4 is a 2D version of step 3. All right, and so once data is in this form, right, it can be analyzed by an algorithm called the fast Fourier transform, which turns the data in step 4 into a frequency plot, as you see in step 5. This frequency plot is the multidimensional representation of the shape that you began with. And this seems very complicated, right, compared to something like area, perimeter, or volume, right? But it's in these little bars here that we find a lot of secrets that are hidden to the human eye. Now, optionally, step 6, we can turn these frequency plots into scalar values, that is, a simple number, right, which is easy for the human mind to understand. We like simple numbers, and it's good for machine learning, right, to do statistics and predictions. And one way to create an index is to just pick 1 bin out of bins, or sum all the bins together into 1 number, right? And so there's more than 1 way of doing that. All right, so that's in general how the algorithm works, right? The core algorithm is ShapeGenie works. Okay, and I want to point something special out about shape, in that even texture, right, or what we call density, has a shape, right? And if you look at these images of microscopic fibers, right, to an untrained eye, right, these might seem haphazard or randomly arranged, right? But it's important to remember, especially in image analysis, that chaotic is not the same as random, because chaos can have recurring microstructure, but randomness doesn't. So one way to quantify an image like this, right, is to measure its texture or its density, right? And so in this case, one way is I just outline the left edge, right, or left or bottom edge of each strand, and I extracted that mask, and now I can overlay the parallel grid system in ShapeGenie, and I can turn this into each of these images into 1 number, and that number is an index of what we call texture or what we call density, right? So it is possible to measure chaotic systems and to find repeatable and objective measures, right, capturing the amount of chaos. This same principle is applied when you measure the turbidity of water, right? How muddy water is, right? Though you can't predict the motion of individual mud particles in the water, you can measure the amount of light that either passes through or does not pass through a vial of water, right? It's called spectral photometry. So we apply the same principle here, right? In a complicated, chaotic system, right? We measure the system as a whole, as opposed to trying to measure individual components. One last thing that makes Shape Genie very, very special, that I'm very proud of, is that we can measure something called pure shape. So if you look on the left side, you have these two objects, right? They might be rocks, they might be spores. They're different shapes and sizes, okay? And if you're measuring area or volume or perimeter, right, you cannot separate area from shape. That is, if at the moment I resize the red object, right, I change its area, I change its perimeter, okay? But in Shape Genie, what you could do is you can normalize both objects to be the same shapes, in this case, the same height, and the height is the the longest internal length, right? And if I normalize them to be the same height while constraining the ratio between width and height, right, otherwise known as an aspect ratio, I'm able to measure shape only, right, without the effect of size. And this has been impossible, right, if you measure area and volume, but now with Shape Genie, it's very possible. And it turns out when we do this for many, many applications, from cell biology to organ biology, this reveals amazing insights, right, that we just couldn't extract before. All right. And so Shape Genie is available. We offer free accounts for students to learn and use. Okay, and you can find tutorials at our website, brainscanology.com/tutorials. Okay, so let's move on to, sort of the more sciencey stuff as opposed to the technical stuff. So why do we study shapes in nature? I probably don't have to say this to this crowd since you're all biologists, but in nature, right, the mantra is that structure is function, meaning if you find recurring patterns of distinct shapes, those shapes will correlate with distinct functions. That's true at the level of molecules, right, and proteins, all the way to cellular structures, all the way to organ structures, right? So structure is function. And so when you find variations of structure, what we call morphology, right, that can be an indication of the presence of subclasses, subgroups, substrains, or subspecies, right, within what you call a homogeneous group, okay? Furthermore, morphology is a proxy of environmental conditions, right? Changes in morphology can reflect soil temperature, soil moisture, right? The moisture of the air, prolonged hot seasons, the presence of pathogens, the presence of pollution. All of these features will affect morphology in one way or another. Okay, and so shape analysis, has the advantage in that all you need is a microscope and a camera, right, and software, as opposed to genomic sequencing, right, RNA, DNA sequencing, which is is more costly. It tells you something different, right? But shape analysis with images is just more cost-effective and easier to do, and it tells you something different than what you know. Okay, so let's now move into the three general categories of mushrooms that I was able to identify. Again, I'm not a mycologist, right? But just for the sake of organization in this talk, I have grouped them into three categories. First is what I would call pedunculated mushrooms, right? That is mushrooms that have a stalk, right, or a leg. And here are some examples of it. And then I found what other people have called shelf mushrooms. That is mushrooms that grow like shelves, bookshelves, but they don't have a stalk. And then you have sessile mushrooms, okay, which don't have a stalk, but they don't grow in shelves. And so I guess this term is already been in use already out there. And there are probably, I found already examples of fungi that are in between these three, right? But for the sake of simplicity, right, I grouped into these three so that I can today just talk about one of them. That is how I would recommend photographing pedunculated mushrooms for shape analysis. And I hope that someone else or other people in this group audience, right, would be willing to create the same recommendations for the other types of mushrooms. All right, so let's just talk, for example, or consider pedunculated mushrooms. So here is a general figure I found online of the general parts of a mushroom. And each of these parts, right, should be compared, should be analyzed on their own, right, relative between different species, right? Because even at the microscopic level, each of these has their own distinct shape features. And today I will just go through a few just as an example. So for pedunculated mushrooms, I would recommend, right, taking these 4 views, right? That is the top view, okay, the bottom view, right, a side view, and then another side view that's a 90-degree turn, right? So if we use the analogy of this person's head, side view would be like this, and then 90-degree turn where the person would be facing you, okay? And the reason I recommend this 90-degree turn is because just based on my examination of images of mushrooms, all of them are not symmetrical all around, right? So there is some asymmetry. And so a 90-degree turn side view will capture some of that natural asymmetry. And when you capture images, it's good to save them as a .tiff or .png file, simply because, I mean, those files are larger in size, but they're larger because they contain more information, right? JPEGs, JPEG or JPG, they're smaller in size, but they lose a lot of information, even though the human eye can't see it, right? And so if you're going to take images for analysis, shape analysis, I recommend saving as a TIFF or a PNG file. So now let's dig a little deeper. Let's talk about the top view, right? So here is this, in this example is, I think this is Amanita muscaria. It's a very popular one, mushroom that pops up on Google when I search for mushroom pictures. And so for this, right, from the top view, right, one thing you can measure is the outer edge, right? It's just the contour of the outer edge, right? But you can also measure, since these scales here have distinct patterns and locations, right? You can extract, and here I just thresholded for these white spots in Fiji or ImageJ, if you're familiar with that software, to get this, right? The mask of these scales. And with the scales, you can measure each shape individually, right? Or you can measure all them all together as a texture. Again, you have to figure out what to do and where the secrets are, okay? And when you have things that are positioned like this, okay, in a radial pattern, right? Oftentimes there might be hidden mathematical patterns there that you can discover if you systematically measure. For example, you might draw radial lines, okay, and only compare the shapes of each particle, right? I guess any particle on the same line, treat it as one group. And so you would have, if you had 360 lines, you would have 360 groups, right? And you can do shape analysis comparing those 360, right? Or you can divide this top view into concentric circles, rings, and then you can group each of the particles according to the rings, right? How far they are out from the center, and then compare the shapes of particles, right? Between groups of rings, right? And you might find a mathematical, right, pattern there that one is just cool because that's how nature works. But two, it might actually help you stratify, whatever substrain you're trying to study or identify. And just as a reminder, why I would recommend these techniques. I mean, look at other cases in nature where a pineapple, right? The scales of a pineapple, they fit the Fibonacci sequence, right? So nature inherently is mathematical. And so even though the scales on this Amanita seem much more chaotic than the scales of a pineapple, that doesn't mean there are not mathematical patterns there. All right, next, let's look at the bottom view. Okay, so the bottom view has a lot of these gills, right? And here I just extracted the outlines of this section of this
video
part of the gill.
You still finished, right?
Right, and then, but for a gill to have radial patterns like this, since it's circular and they all emanate from a center, right? One recommendation would be that you can divide this bottom view into sectors of equal distance, right? Angle distance. And what you could do is you can rotate each of them vertically, right? And then outline what you want to outline and then measure that as a texture. So in that way, when you rotate it vertically, each of these sectors, right, you're objectively comparing each sector in the same way, right? And that might help you make more sense of what's going on here, because there's a lot going on, and to the naked eye, it might seem very homogeneous, as in it all looks the same. But when you measure it, then that's when you find that there are recurring differences. And in radial patterns like this, if you divide them into sectors and measure shape the way we do, you might find that there are different periodicities of shape, right, between different species, right, or between different conditions, right, whether it's nutrient conditions or pathogenic conditions, right, and you only find out if you measure it, okay, in this way. And so again, we know in the study of fruits and plants that nature is inherently mathematical. All right, moving on to the side view, right? So the side view, okay, here I think what might be good to measure is the edge of this cap right here, okay, and also here, and also the top edge of the cap. I think I see distinct textures and shapes there. So those might be good things to measure. Okay, and I also want to point out that stalks themselves can have texture. In this case here, you have smooth parallel ridges in this upper image. In this bottom image, you have very grainy texture, rough texture, right? And texture has a shape in terms of the outline, but also has a shape in terms of the inside. You just have to threshold for color, right, to get the details of the texture within the stalk. And on the point of color, right, we know that when fruits ripen, right, their colors change very systematically, right? And that color intensity can also have a shape. That is, if we extract all the red pixels, okay, from each of these, I guess, stages of tomatoes, right, we will get a shape for the color red, right? So it is possible to threshold for color and get the shape of color. In this case, here's another amanita, and I just did a quick threshold for the reddest pixels, right? And I create a mask of that threshold. This is what red pixels look like, right? And so this is a very messy one, but it's just proof of principle that even color, right, has a shape, okay, or can have a shape. Lastly, so no matter what view, okay, top, bottom, or side view that you are analyzing, mushrooms should be characterized at each stage of development, right? As in they have early stages and then mature stages. And so for every type of mushroom that you're studying, it'd be important to characterize that and do that in a systematic way. And I think since your center is a center of excellence of mycology, you have the staffing and capability to do this on a very large scale, which I think would be highly beneficial to the rest of the field, in mycology. So now I'm going to switch gears a bit and talk about how, right, we're going to go from the macroscopic level of fungi shape, right, to the microscopic level of spore shape. And here I'm going to analyze the spores from Cladonia oculchlora. And when I zoom into these spores, right, I see that they have a general shape that's a teardrop shape. Okay, and they also have a lot of texture. They have rings, right, two inner rings, and then they have a very grainy texture in the middle. And they have this cap at the end here, just translucent cap. So all of these to me are morphology, right? And because I'm a biologist, to me, I always think structure is function. And the moment I see variation, like this one right here has a very pointy cap, while this one has a blunt cap, and this one also has a blunt cap, right? To me, I think subpopulate, okay? And so to start, I just wanted to measure the shape of the outer contour of each spore. So here I just manually trace the outline, all right? You can also do it computationally, right? And in Shape Genie, I then extracted the outline of each of these spores, and then I isolated each of them onto their own canvas, the white canvas. And but before I measured their shape in Shape Genie, what I did was I oriented all the spores such that the tapered end is always at the top, right? In this way, we want to systematically orient all our shapes in the same standardized way. And this provides clean data, right? And we do this because nature is inherently polar, right? As in North Pole versus South Pole, as in anterior versus posterior, right? And dorsal versus ventral. And so when we measure shapes in Shape Genie, if we align the shape according to these natural polarities, the data comes out to be a lot more cleaner, okay? And so I oriented each spore to be like this, and then I did radial grid processing in Shape Genie. And when I did that, I then analyzed, I did principal component analysis, okay, on the first 3 bins of the frequency plot. And I found evidence that there are 3 distinct groups, right? If you look in 3D view, right, these 3 groups separate into their own 3-dimensional space, right? Which suggests that there are, in terms of the shapes, okay, of spores, there are 3 groups that exist in this image. And to the human eye, it's very hard to pick out that there are 3 shapes because the human eye is just very bad at seeing subtle shape differences. But when you do shape analysis, this is the level of detail that you can produce, right? And so I was very surprised and happy that in something seemingly so homogeneous, right, there's actually evidence of 3 distinct morphologies, right? And that's just the shape of the outer contour of the spore. So other shape features that might be of interest to a mycologist in these spores would be the shape of the what I call the cap, right? And here's one way of measuring the cap, the outermost edge of the cap. And to be objective, the blue dotted line stops where at the topmost of the black border on the inside of the spore. Okay, another way is to outline the full shape of the cap, right? And both would work. It's just really up to you as to why you would measure what. All right, and then in terms of the internal structure of the spores, there's just 2 rings, right? 2 black rings you can see. And for here, the image was too fuzzy for me to outline by hand. So I decided to use ImageJ to just threshold. And I was able to, through basic ImageJ features, to isolate the shape of these 2 rings, right, in a way that was objective. Okay, and I would rotate it to be vertical like this and then do radial grid processing. And then what about the inside of the spore, right? There's no obvious shape, but there is an obvious texture, as in there's bright spots and dark spots, right? And so in this case, I cropped out the middle to get rid of the rims. And then I just inverted the image to grayscale and then thresholded for brightness. And I was able to extract a mask of the texture of this inner region, right? And so for something like this, I think the radial parallel grid processing system would be a good choice. Again, both processing systems work, whether radial or parallel. Those are 2 options ShapeGenie gives you. You have to figure out which one gives you cleaner data. But all grid systems work. Just some work better than other. It depends on the shapes you're working with. And so that's the end of my presentation. I wanted to leave time for some question and answers. I'm sure you have many comments to make, since I'm not a mycologist and maybe some of the stuff I said is just completely wrong. Please forgive me. But yeah, I would like to open the floor now to take any questions.
Hello, do anyone have question?
I have one question that may you suggest about if you want to take a photo about the mushroom that produce of the many pore, like a polypore that's produce many pore of the circle shape or some irregular shape. How to do it better? Yes, based on this.
Wow. So you're asking how do you take pictures of spores that come from mushroom? That's a good question. I mean, I don't know. The spore image, I took that from online. There's a French mycologist who posts beautiful pictures. So I analyze his pictures. But I would imagine that if it's airborne, right, you can tap it onto a glass slide, right? And then, the conventional glass slide, with a liquid solvent that might solidify over time, right? Like a resin, right? That would capture it, and freeze it in time. Mm-hmm.
Yeah. Do some students have more questions?
Oh, somebody have a question? Well, I have a question. I guess I'd like to hear, I guess somebody want to volunteer and tell me about your research, since there's so many of you here. And, I just like to learn and see what questions you're working on. Is that Nagme?
Since nobody is giving an answer, I will briefly tell you what some of the research we usually do in the center. So we have groups that look at basidiomycete mushroom, obviously, and we have some group which look at ascomycetes, but we also look at endophyte, pathogen, subprobe. Most of the study is either subprobe or pathogen. So if there was any way we could, for example, screen the spores that we can get from the pathogen, so that would be quite interesting to do, because this would give us an idea on a large scale what is the diversity of some of the fungi that we have in nature. Because the problem we usually have is many of the fungi we get, we cannot culture them. So just we can observe them, but we cannot culture, because the culture is very important for next application for all these things. So having an idea what we are surrounded by would really give us, yes, it would be very interesting to know, but not sure how.
Oh yeah, I mean, that sounds like a great question that you're working on. I mean, for that, yeah, I think shape analysis would be very helpful there, just based on, you know, I've looked at many, I guess, several types of spores, right? From this one French mycologist who posts pictures of spores. And so there's a lot of recurring variations, but I mean, just like an example I gave, you know, I'd be happy to do a pilot study with you and just prove to you, right, analyze a few groups of spores that, you know, what we can find. And anything that we can measure precisely, we can, in terms of shapes, right, images, we can develop a machine learning model that can quickly classify it, meaning you can just take a picture of a spore, right, load it to the model will spit out what percent it is basidiomycetes, what percent it is pathogenic, or whatever the categories are. That's very doable, right? I mean, we have the machine learning algorithms already in place. And the only challenge is how to extract information in a way that helps you find unknown subtypes, right? Because right now, for image analysis, image classification, deep learning is a very popular AI tool, right? And deep learning is, you know, and for images, it has to do with convolutional neural networks, right? CNNs. But the problem with deep learning is that it doesn't tell you why it made that decision, right? And it can't find subtypes, right? If you tell it there's only 3 groups, it only finds 3 groups no matter what, because you told it. Okay. So our software allows you, because we measure things so precisely, and we allow the expert to dictate what to measure and why, right? Based on your expertise, we can find gradients, right? So we can find subtypes. And once you find the subtypes, then you can train deep learning to find the subtypes, right? And so, you know, Shape Genie, it can work hand in hand with deep learning, right? You know, they're not sort of opposite of each other, right? They're just really good at different things. You know, for Shape Genie is great for discovery, and deep learning is great for speed, right? Once you know what the categories are and how to define them, then, you know, deep learning is great for speed.
Thank you. Thank you so much. The only thing I can think of for now is where we might get stuck is, for example, we have many species which can be, for example, endophyte, subprobe, and pathogen, and the spore is the same between all the different lifestyle. So that might be a challenge for us later to try to solve. This is not really for now, and many different genera. So if we try to go general level, like lower level, try to identify the species a bit closer to what they might be, this might bring another problem where many different genera have very similar spores. So this might give us another thing to think about. But thank you very much for sharing this software with us. Is very interesting to us, and I hope we can work on something in the future. So thank you very much on behalf of all of us.
I'm very happy and feel privileged to be here. You guys sounds like a great research center, and I had no idea until now. So I'd be happy to help you. Looks like we have a question. We have a hand raised from Carlo Chris Apurio.
Hi, Dr. Dave. I'm actually very interested with the talk. If Dr. Avi was actually looking at it at a very macro perspective, I want to bring it down to a more micro perspective also, because like I could see also the potential of using the software in like differentiating specific, because sometimes we're looking at spores which look almost the same to the human eye, but it can actually have differences, and I am seeing the potential of using the software there. You were asking earlier, how do you declare a new species when they actually look like the same to me?
Yeah.
We usually use the molecular data for that, but we also need to see some differences in the morphology. And I think that is where the software can help us. I think that's one thing that I want to confirm. And secondly, in using the software, like what you showed earlier, we need to use masking in order for it to be analyzed. Is the masking like part of the software, or do we need to use to do the masking manually and then place the masked image now into the software? Because I think there are still many variations that can happen in the masking. For example, it may not be accurate and there might be errors in the analysis because of wrong masking that will happen. So I'm interested if that is integrated into the software or that is done separately. Thank
That's
you.
great. Yeah, that's a great question. Thanks for asking that. In terms of masking, right now we have two. The simplest option is to do a manual tracing, right? Because that allows the expert to, be very precise in what they outline. And the tool we created to make manual tracing easy is that we'll give you 4 colors to pick from, right? Bright red, bright green, bright blue, and bright magenta. Okay, and you can outline on your image. Okay, and once you upload that image with the outline, we have a tool that extracts that color, or those 4 colors out. So we automatically create the mask for you, right? So that's the tool we create to make manual tracing easier. But the second way that's more effective as possible is just thresholding, right? Through color, right, or intensity. Okay, and that is, you filter for the brightest pixels, right, of certain color channels, and therefore you can create mask of the every particle at once in your image, right? And that is done right now. We do through PG or ImageJ, which is open source software, right? So it's free. But in the future, we'll build that into our own Shape Genie. But for now, the beta version, we don't have that yet. So we recommend using something like ImageJ or Fiji. And lastly, if you know exactly what you want to segment, right, or trace and why, then you can build an instance segmentation model, right, through TensorFlow and Python. There's many ways, right? There's Mona AI or Mon AI, right, from NVIDIA. But Python has PyTorch and has TensorFlow, and that's what we do. We build it. We label, like for brain scans, right? We label parts of the brain in the MRI, and then we train it to pick out those shapes, right? And when we label enough data over time, it becomes very accurate, right? It's like 90-some% accurate in picking out that shape. And so that quickly, every time we upload a MRI image, right, it picks out that shape right away, right? And so that's what we're doing to speed things up. But when you're first starting out, unless you know exactly what you want to measure and why, it's probably not a good use of your time to build an instant segmentation model right away, because it's a lot of labeling, right? And you have to know exactly what you want to measure, because it only measures what you told it to measure, right? And so, yeah. And so typically for pilot studies, we do the manual method, but then there are thresholding methods that are faster, and ultimately fastest is a segmentation AI.
Thank you very much. So that means that there is really a big potential, right? Because if we start feeding data to this software, like what you said, we can use AI in the future to do a very macro type of a very big study on the data from machines. So thank you very much, Dr. Dave, for this very interesting talk.
Yeah, well, you're welcome. Yeah, so anything that you can turn into a number that you can do statistics on, right? That has potential to develop an AI model on, right? Because AI is just, statistics, right, with computer together, right? That's what it is, right? Very advanced statistical models at the speed of computers. So yeah, anything that you can do statistics on, you can turn into a number, you can develop a machine learning model for, and develop AI for.
one question in the box chat.
Okay, yes. Somebody named Pranami Abe Wikrama asks, hi, Dr. Dave. Thank you very much for the nice talk. I'm really interested about you're saying that color has a shape. Do you think that we can use software to measure disease symptoms? Please. Absolutely. Absolutely. Yeah. Yeah. Leaf spots, they have a shape too, right? They're very globular or globular. And so it's hard for the human eye to objectively identify them or quantify them. If you try to measure something like area, it'll give you very noisy data, right? Because two blobs that have the same area can have very different shapes, right? But yeah, definitely Shape Genie can measure blobs very precisely. You know, I hope you apply it to studying leaf spots. And so where do you, I got a question for you while you guys are thinking of questions. I guess where did most of your students come from? Or your scientists?
So it's like we have students from 9 different countries.
Mm-hmm.
So we have students from China, Sri Lanka, Iran, Thailand, Myanmar, Laos, Philippines, Mauritius. I think I missed one.
That's great. Yeah, that's
Yeah.
awesome. Yeah, I'm you know, my family ancestry is Vietnamese, but my wife is from Iran. She's Persian. And so it's great to see you know, such I guess diversity in your center. Yeah, maybe I'll visit one day. Sounds like Thailand's
You're
a
welcome
place
to
that,
come and visit us.
yeah.
Oh, yes, India as well. Yeah, so yeah. So we have from India also. So if there are no further questions, can we go back
Okay.
to the presentation? Okay.
Oh, you want to see the slides again, or what do you mean? Or I'm done, really. I mean, this last part is
Okay, so this is only a discussion, right? So the students, do you have any more questions? Students or researchers or lecturers, do you have any more questions to ask? No questions? Gig?
No more questions.
Okay.
Okay, if we don't have any more questions, let's finish this webinar.
Okay, well, thank you very much for the invitation, Roxine, and for organizing it. I will send my slides and recording, which will be posted online for you all. Yeah, so I'll be in touch about collaborations, and feel free to reach out to me if you have questions about doing shape analysis on your research.
Okay, so thank you very much, Dr. Dave. And thank you everyone for attending today's webinar. Thank you, everyone. Thank you, Dr. Dave, again.
And bye-bye for now.
Bye-bye.
Thank you.
Thank you very much.
Bye bye.